Well, ladies and gentlemen, good morning, and Munir, thank you very much for the invite. So I thought I'd give this talk the title that's sort of what we've been trying to do, and that is form partnerships with the pharmaceutical industry, try and exploit genetic insights, try and exploit patient resources to try and accelerate the generation of new therapeutics for patients. Now, I'm interested in the discovery of new medicines. I believe the things I worry about, I worry that as a biomedical community, as an industry, we're not producing enough new medicines. I'm increasingly worried that when we do produce a new medicine, frankly, it's becoming increasingly unaffordable, even in the UK. And that situation is just going to get worse and worse. I'm also worried that within biomedicine, biomedical research, there is a lot of duplication. Many companies work on the same few ideas in parallel, in secret, and of course most of them fail. I'm also increasingly worried that many of our pharmaceutical scientists keep telling us that 50, 60, 70% of academic literature, they can't reproduce it. We need to do something about it. There's no point getting defensive about it. We need to do something about it. But let me just kick off and let's just think about what patients want from us. What does society expect from us? What do their carers, their relatives, their families expect from us? I think what they want is novel medicines. They want effective medicines. They want affordable medicines, and they want them quickly. Let me just share a few figures. So I said novel medicines. We need them in so many disease areas. Take cancer. In the next 12 months in the UK, 350,000 people will get diagnosed with cancer. Half of us in this room during our lifetime will have a diagnosis of cancer. In the next 12 months, 14 million people on this planet will get diagnosed with cancer. We need new treatments. I worry a lot about dementia. In the UK today, we've got 850,000 people with dementia. In 2050, that's going to be 2.1 million people, 2 cities the size of Birmingham. All of us who live to more than 80, 1 in 6 of us will get it. The cost today of looking after a dementia patient in the UK is £32,000 a year. If you add all that up, we're spending £26 billion a year looking after our dementia patients. 26 billion, incidentally, is if you add up all of our household energy bills, that's 26 billion. That's how much we're spending. And we've not had a new treatment for dementia since 2002, and that treatment only works in the first 9 to 12 months after diagnosis. I worry about mental health. You know, the figures across Europe, 83 million people have got some sort of mental health condition. 25% of people during their lifetime will have some sort of mental health condition. Anorexia. Take 15-year-old girls in the UK, out of every 150, one of them has got anorexia. Rare diseases. If you add up all these rare diseases, there's 7,000 rare diseases. If you add up all the patients, that's 350 million patients on the planet. 95% of those patients have absolutely no treatment. Sally Davis has talked about antimicrobial resistance. Today, 700,000 people a year are dying because they're resistant to current antibiotics. In 2050, that figure is going to be 10 million. We need lots of new drugs. I said we need effective drugs, and you might think, well, why is he saying that? Aren't all the drugs that are out there effective? Well, 3 years ago, there was a publication where these authors looked at solid tumors, the treatments for solid tumors. At the time, we had 71 treatments for solid tumors, and they looked to see what was the average increase in progression-free survival or overall survival with these treatments. The average increase in progression-free survival was 2.5 months. Overall survival, 2.1 months. And of those 71 drugs, they concluded that actually only 30 of them had clinically meaningful efficacy. So 71 drugs out there, but only 30 of them have clinically meaningful efficacy. I said we need more affordable drugs. Again, 4 years ago, there was a publication where they looked at, in the UK, in the year 2000, we had 69 drugs for treating cancer. Of those 69, 50 of them were cytotoxic agents. The average duration of treatment was 181 days. The average cost of the treatment was £3,000. And in the year 2000, £3,000 equated to 20% of our GDP per capita. They did the same analysis in 2013. Now we had a further 63 drugs for treating cancer. The average duration of treatment was now 263 days. So it's gone up from 181 to 263. And the average cost of that treatment was now £35,000. It's gone up from 3,000 to 35,000. And 35,000 equated to 120% GDP per capita. So it's gone up from 20% to 120%. And that trend is continuing. I said we need drugs quickly. Of course, for patients, even tomorrow is too late. They want it today. So we need novel drugs, we need effective drugs, we need affordable drugs, and we need them quickly. Now, I believe the way we're currently doing drug discovery, it's incredibly inefficient. It's too costly, it's too risky, and it's too slow. There's lots of reasons for that. I think many diseases, we don't understand them well enough. We don't really understand the molecular causes of schizophrenia or depression. A lot of the animal models that we use do not predict what happens in the clinic. For many diseases, we do not have good biomarkers. And there are many drugs out there, we don't even know what their molecular mode of action is. So if we don't know how existing drugs work, like paracetamol, how are we going to design better drugs? Some major scientific challenges. So let me share with you what we've been trying to do over the past decade in Oxford. So we've tried to do 4 things. Firstly, we've decided to try and pool resources to try and share risk. So currently we're working with 9 large pharmaceutical companies. Each of these companies is giving us 5 million euros over 5 years. So we have more than 40 million euros of private funding. We've got funding from 7 patient organisations. The Wellcome Trust has put close to £60 million into our lab in Oxford. We're pooling all of these resources to try and share risk. The second thing we do is that we only work on completely novel genes, novel proteins that we think could be drug targets of the future. We don't work on genes or proteins or targets where there's already 1,000 publications and 50 labs working on it. We only work on completely novel genes. And for those genes, what we do is we purify the human protein. I'm not interested in mice and rats. We purify the human protein. We build biophysical, biochemical assays. We work out the 3-dimensional X-ray structure. We generate small molecule inhibitors. We generate antibodies. Now, these reagents are really high quality. They're high quality not because we're smart. They're high quality because we're tapping into the resources of these 9 large pharmaceutical companies. We get access to their compound collections, their medicinal chemistry, how to develop cell assays, do screening, etc., etc. So by working on novel genes and generating novel tools, we're trying to drive innovation. The third thing we do is that all of these reagents, which are starting points for drug discovery, we make them freely available to the world. We give them away to anybody in academia, anybody in biotech, and anybody in pharma, because I believe that's the best thing I can do to facilitate science and therefore facilitate drug discovery. Now, of course, the consequence of that is you can imagine every academic who comes into my office wants to collaborate with us. They know I've got no secrets. We'll share all of our know-how, all of our expertise, all of our reagents. That transparency creates a lot of trust, which is great for collaboration, it's great for science, and it's great for drug discovery. So we're now working with more than 300 academic labs all over the world. And the fourth thing we do is that all of our data, all of our reagents, all of the knowledge that we generate, we release it immediately. We don't sit on it for 12 months while we're writing up a manuscript, because in those 12 months, there'll be people out there wasting their time and resource. So 4 things. Pool resources to share risk, work in novel, impossible areas to drive innovation, make everything freely available to crowdsource science, and release everything immediately to reduce duplication and wastage. So what's been the impact of this? So let me share with you one example. So when I went back to Oxford in 2008, the Wellcome Trust gave us some money to generate novel inhibitors for novel epigenetic proteins. And we can go into a long lecture about why epigenetic proteins, but we decided to work on a couple of families of proteins. One of them was bromodomain proteins. Now, many people said to me then, 2008, Chaz, you'll never generate inhibitors for bromodomain proteins because it's a protein-protein interaction, it's impossible. Well, we worked on it. We worked with GSK, and we worked with Jay Bradner at Harvard at the time to generate what was the first novel inhibitor for a bromodomain protein. It inhibited a protein called BRD4. Now, this inhibitor, it was potent, it was selective, it was cell active. So we thought, okay, right now we've got this novel inhibitor. Well, what does it do? At the time, there were 2 publications on BRD4. One of these publications said that when the BRD4 protein is fused with a protein called NUT, those individuals develop a rare cancer called NUT midline carcinoma. It's such a rare cancer, most oncologists can't even diagnose it. Now, Jay Bradner, working at the Dana-Farber at the time, had access to patients with NUT midline carcinoma. So what he did was he took these patient-derived cancer cells, he put this inhibitor on those cells, he showed that it reduced proliferation, it increased apoptosis, and when he took these cancer cells and he put them into a mouse, the cells multiplied, the tumor got bigger and bigger, and when he dosed the inhibitor, it stopped the tumor growing. So we published all of that data in Nature in December 2010. That's 9 years ago. That molecule now, Jay Bradner, GSK, ourselves, we've given it out to several thousand labs across the planet. And these labs have taken that inhibitor and shown that it works in a whole range of cancers. There's data in sepsis, in fibrosis, cardiac hypertrophy, and there's even a publication in male contraception, and there's probably other publications that I don't know about. This is crowdsourcing science. There are now several thousand publications using that inhibitor. So we've facilitated academic science. We've also enabled proprietary programs in the pharmaceutical industry. So the pharmaceutical industry has looked at these thousands of publications and started their own program inside to generate their own inhibitor with intellectual property, etc. Today, there are more than 20 companies doing more than 30 clinical studies with their own BRD4 inhibitors. So we've also accelerated translational studies. On top of that, in 2011, Jay Bradner set up a company in Boston to take that inhibitor all the way to the clinic. He called this company Tensure Therapeutics. He got $15 million of VC funding to set that company up. And then in January 2016, Roche bought that company for $535 million, more than $500 million. So we've also enabled the creation of new companies and new jobs. That was the first inhibitor we've generated. Today, we've generated more than 70 of these inhibitors, and they're all freely available to anybody who wants to use them. What have we been doing more recently? More recently, what we've been doing is we've been working with lots of clinicians and geneticists and disease experts to try and identify high-priority novel genes which they think could be drug targets in various diseases. So we're working in cancer, metabolic diseases, inflammatory diseases, and neuropsychiatric diseases. And what we do is we take these genes and we do what we're good at. We make the protein, we develop assays, we do the structure, we generate chemical starting points. This package of reagents we call a target enabling package. And we've generated now more than 20 of these packages, and we believe that these will enable translational studies both in academia and in industry. What we've also been doing is, of course, sitting in a place like Oxford, we're surrounded by hospitals, we have access to lots of clinicians, lots of patients, and lots of patient material. Now, as I said earlier, I'm not a fan of animal models. I think largely they're a waste of time. I would much rather take disease cells from patients and study our inhibitors on those disease cells. And so that's what we're doing. And of course, in two areas in particular, we can readily access patient-derived cells, and one is cancer, the other is inflammation. And we're setting up platforms where we can routinely screen our inhibitors through lots of different types of cancer cells and lots of different types of immune cells. I think that's a much better way to come up with new targets. We're now cooking up a couple of other plans. So one big project, with Munir's help, we're trying to set up a national effort to accelerate new therapeutics for multiple morbidities associated with aging. So we recognize elderly patients don't have one disease, they usually have half a dozen diseases. We now believe that there are certain targets and pathways that we can go after which will treat multiple morbidities associated with aging. And so we want to take some of these novel ideas, generate industry-standard clinical molecules, and we want to test those molecules in patient studies in the NHS. So we're setting up this national effort involving the University of Liverpool, Dundee, Birmingham, Oxford, the Crick, the Medicines Discovery Catapult in Manchester. And the second project we're trying to do now is we're going to take some of the genetic outputs of the Genome England project, where they've identified clear linkages with specific clinical phenotypes, and we're going to take those genes and we're going to generate these target-enabling packages for those. And we think, and as with everything, we'll make it all freely available to try and accelerate science. So to sum up, I believe that in early discovery, there is too much secrecy, there's too much competition, there's too much duplication, there's too much wastage. What we're trying to do is bring together lots of clinicians, lots of pharma companies, lots of academics, biotechs, CROs, patient groups. We're trying to work together to come up with novel targets for drug discovery, novel clinically validated targets for drug discovery. We think that's good for industry, we believe it's good for patients, and of course it's good for the economy. What we're trying to do is to accelerate more novel treatments for patients, more effective treatments for patients, more quickly. And I hope, ladies and gentlemen, that these treatments will also be more affordable. Thank you very much.